Subscribe this channel by pressing this button for being updated from Biomedical and Healthcare Society.
Hello guys, welcome back to the channel. This is the second chapter, Chemical and Enzyme-based Biosensors from the lecture series, Biosensors and Transducers. So without any further delay, let's get started. The agenda of this chapter includes the introduction to the chemical and enzyme biosensors. Then after that we will study about the blood gas sensors. Then we will study about the blood pH sensor. After that we will study about the PCO2 sensor. After that we will gonna see about the Microbial biosensors. At last we will see about the enzyme-based biosensors. So let's start with the introduction. In the previous chapters we have learned about the sensors and transducers, and as per the definition we have learned, we got to know that a sensor is a converter. It's a type of a converter that measures a physical quantity and converts it into a signal which can be read by an observer or by an instrument. Also, we can say that a sensor is a device which senses the signal or any physical change happening in the instrument. That instrument can be termed as the human body in the case of leading towards the biomedical equipments. A biosensor is an analytical device that is used for the detection of analytes that combines a biological component with a physiochemical detector. Now also if you see more about the biosensor, this device incorporates a biological sensing element that is either intimately connected or integrated with the transducers. We all know that transducer is a device which converts non-readable quantity to the readable one, that is, which changes the one form of energy signal to the another form to make that form of energy into the readable one. Okay, after that, this recognition based on the affinity between the complementary structure, which is like the enzyme substrate, antibody antigen, receptor hormone complex are being involved into it. The selectivity and specificity depends on the biological recognition system which is connected to the suitable transducer. It is basically the analytical device that will directly convert a biological response into the electrical signal, which detects, records, and transmits the information regarding a physiological change or a process. Also, it determines the presence and concentration of a specific substance in the test solution. Suppose we take the example of a glucose strip. we have added the blood in the glucose strip and we are measuring the glucose. That glucose strip will measure the amount of glucose present in the blood and give you the reading, and as per that reading you will get to know that whether that glucose concentration in the blood is normal or not. The basic principle of biosensor involves the implementation of the three elements. The very first element considered as the biological recognition element, which is highly specific towards the biological material analytes produces. That the analyte is producing the biological material which is being recognized by the biological recognition element. The second element is the transducers that detects and transduces the signal from the biological target receptor to the electrical signal which is as per the reaction is occurring over there. The third element which is being implemented as a principle of the biosensor is the transduction signal which is coming from the biological recognition element via the transducer and converted to the electrical signal which is being further amplified as per the requirement and being read out by the detector after the processing the values which is being displayed from the monitor and the controlling system. This is the basic block diagrams of the biosensor. You can clearly see the very first element we are having the analyte. Analyte is being added and reacted with the substrate and gives some further changes which is being intimated and recognized by the bioreceptors, which is further went to the molecular recognition, and that change is being read by the transducers and converted into electrical change signal, that is unreadable quantity to the readable one, and further that is being transferred to the measurement section that is in the processor to measure it, to amplify it, to filter it, so that we can clearly understand which type of signal we are getting and how that signal is, and that data, that signal data that we have got from the transducer after the transduction process is done is further transmitted to the data recording center, that is the display and the data memory center, where this memory is there and in that memory the data is being stored. The biological material is immobilized and contact is made between the immobilized biological material and the transducer. The analyte binds to the biological material to form a bound analytes, which in turn produces the electronic that can be measured. Once the analyte is bonded with the substrate, it produces some changes. It shows some changes. As we all know, when the substrate is bounded with the analyte, the substrate is taking the shape of analyte, and as per that it accepts the changes. It shows the variation in the analytes. Sometimes the analyte is converted to a product which could be associated with the release of heat, gas, electrons, or hydrogen ions. These are the changes which is being occurred when the substrate is bounded with the analytes, and these changes being identified by the help of bioreceptors. The transducer then converts that product linked to the changes into the electrical signal, which is further been amplified and measured. Now let's see the components of biosensor. Very left side of this you can see the circular, triangular and square shaped elements in different colors coming towards one orange color element. Those are the substrate which is been binding with the analytes, and after the binding you can see in the next block the bioreceptor is there. Below the bioreceptor you can see the molecular recognition material is been stripped in that block, and in that molecular recognition material we can clearly see there are some names been written. That names are the changes which occurred by the binding of the substrate to the analyte, named as the enzyme, antibody, microorganism, CDL, etc. After that, that changes is showing some property like electroactive substances, pH change, heat change, a light change, mass change. These are the changes which been shown by the bonding of the substrate with the analytes on the bioreceptor panel. After these changes in occurred, that change been identified by the transducer, I mean to say that respective transducers, and that change is being further transferred and changed to the signals with the help of the respective transducer like electrode, semiconductor pH electrode, thermistor, photon counter, piezoelectric device, and the signal is being converted into the measurable signal. That measurable signal is further transmitted and converted to the electrical signal quantity, and at last it is sent to the detector. In that, very first element is the biological element. The component used to bind the target molecule, which must be very highly specific, stable, under storage conditions and immobilized. You can see the substrate, active site and the enzyme in the diagram. as per the binding is happening in the biological element with the substrate to the enzyme, there are the changes occurring like microorganism, tissue, cell, organelle, nucleic acid, enzyme component, receptor, antibodies. These are all comes into the biological elements, which is showing some kind of change after the binding of the substrate and enzymes. The function of the biological element is to interact specifically with a target compound. That is, the compound is to be detected. It must be capable of detecting the presence of a target compound in the test solution. That is, I mean to say that the biological element is that much capable, it should detect the presence of the targeted compound which is present in the test solution. The ability of a bioelement is that it has to interact spatially with the target compound, which is the basis for the biosensor. So from this we can understand the working mechanism, how the biological element is important in the biosensor and how it is working. The second and very most commonly used and important element is the physiochemical transducer in the biosensor. This physiochemical transducer acts as an interference that measures the physical change which occurs with the reaction site at the bioreceptors and transforms that energy into the measurable electrical output. As we all know, this transducer work is to convert non-readable quantity to the readable one, that is the energy, unreadable energy to the measurable readable energy in the output form. So in this block we can clearly see electroactive substrate, pH change, heat, light, mass change. These are the changes which being seen at the bioreceptor sites after the bio element is been added, and these changes being measured with the respective physiochemical transducers like electrode, semiconductor pH electrode, thermistor, photon counter and piezoelectrical device respectively. The third and the very most important component is the detector. Detector will detect the signal, measurable signal which is coming from the transducer, or we can say whatever the signal is coming from the transducer further being passed to the microprocessor where it is being properly analyzed, amplified, filtered out, and that data is further converted to the required units concentrated with the proper alignment of the data and transferred to the display and the storage unit. This is the particularly specified task for the detector. The principle of detection involves various transducer use, and as per the change this detection occurs. In the very first, we can say that the piezoelectric change detection, or I will say, that is being done with the help of the measured change in the mass. Whenever we will see the change in mass, then we will use the piezoelectric transducer for measurement of that change, for the measurement of that detection. Whenever we see the change in the electric distribution, or if we want to measure the change in electric distribution, we will go for the electromechanical transducer that will help to measure the change in electric distribution. Similarly, when we want to detect the change in light intensity, we will use the optical transducer, and that optical transducer will be used for the detection of the change in light. Parallelly, if you want to measure the change in heat, then you have to use the calorimetric transducer. If you use a calorimetric transducer, then you can easily able to detect and measure the change in heat. Now let's see the working of biosensor. In this diagram you can see that the naming is being done like A, B, C, D, E. Those are the name of the elements taken in the diagram. Like A is the bioelement, B is the transducer, C is the amplifier, D is the processor, and E is the display. As you can see over here, that S and P, that is a substrate, is being added with the analyte, and some physiochemical changes being shown in the bioelement, that is the A panel. That change is being analyzed and being recorded by the transducer and converted into the readable form. That is, that physiochemical change is being converted to the measurable change in the form of signal, and that signal via the transducer is further transmitted to the amplifier, where that signal is being amplified up to the extent which is required to be measured by the processor. Then that measurable signal and amplified one is being transmitted to the processor named as D, and that processor will have the logic circuits where it will check whether that signal is appropriate or not, and if the filtering is required, it will do the filtering, and some modification is required, and that modification will be taken care, and after that, that is being further transmitted to the display unit, that is consisting of the element name E. So this is the basic schematic diagram of biosensor, how the working of the biosensor is going on. First, A, bioelement, second, B, transducer, third, amplifier, processor, and display. This is the chronological chart of working of biosensor. Basically, the biosensor involves the quantitative analysis of various substances. By converting their biological actions into measurable signals, we can clearly measure and analyze the signal of the which is being observed by the biosensor from the biological, or we can say physiochemical, changes involved element. Generally, the performance of the biosensor is mostly dependent on the specificity and sensitivity of the biological reaction which is occurring at the biological element by the addition of the analyte and substrate involves the physiochemical change shown at the biological element, which decides the stability of the enzyme. Similarly, as per we have seen that schematic block diagram, we can see one more diagram over here for the working of biosensor. You can see on the very right side we are having the analyte which is been added with the substrates in the form of sample handles and preparation. How we are preparing the sample? By the addition of the substrate to the analyte, which gives you the biological change and physiochemical reactions occurred at the biological element, and that change is being further detected and transmitted by conversion with the help of transducer to the measurable signal, and that signal is been further transmitted for the analysis, which we can see in the graph. And as per that analysis, the response is being figured out by the processor, and that response been transmitted back. These are the some characteristics of the ideal biosensor. Very first in that we are having the output signal must be very much relevant to the measurement environment. That is the measurement environment which we are using, the signal which we are getting in the form of output, that should be relevant to that. The functional surface must be very much compatible with the transducer. What if the transducer is not compatible with the functional surface? Then the transducer will not be able to measure that signal properly and give you the results properly. In terms of the functional surface, it is required to, the compatibility should be there. The biosensor should be highly specific and selective in nature in terms of the detection of the biochemical change, or we can say the physiochemical change involved with the help of the analytes and substrates we are getting into it. It is sufficient sensitivity and resolution involved with the transducer while measuring this physiochemical change. The sensitivity should be high and the resolution should be at up to the marks to measure that change properly. Parallelly, as per we have seen the characteristic in the previous slide, furthermore characteristics are there. It should be sufficiently accurate and repeatability measurement should be there. The speed of response should be sufficient. That is, it should not have a lot of delay involved in that response. It should be like in very less, it should take very less amount of time to detect it and to give the output. It should have the sufficient dynamic range involved in the measurement and giving the response of the physiochemical change occurred in the biosensor. Insensitivity to the environmental interface, or we can say their effects must be compensated. That is, the external element which are being present in the environment, it should not affect the biosensor. So these were some of the characteristics of the ideal biosensor. Now let's see the basic characteristic which is very much required in any biosensor. If you talk about, it should have the property of linearity. That is, the sensor should be high for the detection of the highly substrate concentration present in the bioreceptors. The sensor should be designed like that. It should have the sensitivity value very high so that the electrode response per substrate concentration should be high. The selectivity should be very good. That is, the chemical interference whatever present over there must be minimized for obtaining the correct result. The response time should be more than 95%. Why it is required? Because if the response time is low, we will not be able to get the real-time analysis of the data of that signal which is giving you the physiochemical change involved in the subtraction of the involvement of the substrate and analyte, giving you the response in the biosensor. Now we are going to see about the blood gas sensors. As per the name suggests, blood gas sensor, that is, we gonna see about the involvement of the gases which are present in the blood, and as per that we are having some sensor to measure the gases. Okay. In that, the analysis of blood gas values have been accomplished via the electrochemical device commonly referred as electrodes. All traditional electrodes which have been already there present in the scenarios use the biological blood gas analysis to measure changes in either electrical current or voltage and equate these changes with chemical measurements. Moreover, recently the new techniques are opted, such as optodes, which has been used to determine the blood gas values, and this is very much accurate as compared to the traditional electrodes methods which have already been compromised for the measurement of blood gas. Very few primarily used blood gas electrodes are present are there. In that we are having three electrodes. Very first in that we are having the pH electrode, second one is the PCO2 electrode, and the third one is the PO2 electrode. These three electrodes are primary electrodes and used for the blood gas measurement. And in the diagram we can clearly see that two charcoal colored like cells are present. The very first is that the reference half cell, another one is the working half cell. Below that we have the electrolyte solution, and as per the potential difference occurring over there, and we are getting the electrical measurement device, and as per the electric changes are occurring in the electrolyte solution, the electrical measurement device give you the change in concentration of the various things, and as per that change in concentration of various electrolytes present in that, we are getting to measure the change in concentration of the solution present in the electrode. So very first we will see the electrode function in the blood pH sensor, that is one of the type of the blood gas sensor. So the pH electrodes differs greatly from the PO2 electrodes because both are having the same structure and same function, but the electrode's quality and the selectivity is different as compared to the PO2 electrode. Functionally wise, the pH electrode measures the change in voltage rather than actual electrical current. Specifically, the pH is measured by the potentiometric method, by which an unknown voltage is measured by comparison with a known voltage and is then converted to a pH. To accomplish this, the pH electrode requires 4 electrode terminals instead of just 2, such as which are already present in the PO2 electrode. When we gonna see the PO2 electrode, in that we will be having basically the 4 electrode terminals. Majorly they are using nowadays 2 electrode terminals, but in traditional methods they were having the 4 electrode terminals. The functionality of the pH electrode and the PO2 electrode both are same, the structure are same, the mechanism is same, just electrode quality is different, and that has been calibrated for measurement of the pH level and pH type, and that is for calibrated and designed for the measurement of oxygen. So in that, following up, we are having a reference solution which is known as pH, and it plays in between the 2 terminals, which having one is a reference voltage to compare the unknown voltage in against of that. If you see in the diagram, left side we are having the half cell of AgCl, and right side we are having the half cell of HgCl. The fluid is having the unknown pH on the right side. On the left side we are having the reference solution with the known pH. Okay, the voltmeter has been added. We are having the common pH sensitive glass electrode terminal, which will attract the H plus ions toward itself when the voltage is been implemented in the fluid. As per the change of the voltage and as per the addition of the H plus ion and implementation of the voltage and the attraction of the H plus ion to the glass electrode terminal, the change in the voltmeter readings will be there, and that change in the voltmeter reading will be amplified and further be measured to get the data of pH. Or we can say a single unique pH sensitive glass electrode is the terminal services is used as the common electrode terminal for the both the reference solution and the pH solution, unknown pH solution also, and that reference electrode, or we will say that pH sensitive glass electrode, will give you the data of the presence of the pH in that solution. This blood pH sensor specifically manufactured pH sensitive glass allows hydrogen ions to diffuse into it in proportion to the hydrogen ion concentration of the fluid which is to be exposed in the solution. Because of the different solution on either side of the glass, a net electrical potential develops between the two fluids and is quantitative at the voltmeter reading, and that relationship between the voltage and the pH at a particular temperature is described by the modified Nernst equation. In general, for each pH unit, difference between the known and unknown solution, a difference of around 61.5 millivolt is being developed. A specific voltmeter converts voltage to pH units based on the Nernst equation and visually displays the pH. The physical component which is present in the blood pH sensor that involves the system which will decide, or we can say which will divide physically into two major components. One component is the pH sensitive glass at the tip and is the silver chloride half cell inside it. Because this component is the site at which the actual blood comes in a contact with the outer surface of the pH sensitive glass. It may be referred as the working half cell. Thus the mercurous chloride component can be called as the reference half cell. In the diagram we can clearly see left one is having the reference called as the reference half cell, and which the mercurous chloride is been added with the KCl and liquid junction into it. On the right side of the diagram we are having the working half cell in which AgCl chloride, silver chloride is been implemented in that with the inner portion of the inner electrode, and which is known to the reference solution and added with the pH sensitive glass, and in that beaker this solution is being added. The pH sensitive glass component is often referred to as the working of the measuring electrode, and the other component is called a reference electrode. The reference half cell includes an electrode terminal made of mercury coated with mercurous chloride. The term calomel is often used for this type of electrode terminal. The calomel electrode terminal interface with a platinum wire that transmits the change in voltage to the voltmeter, and the relation between the voltmeter reading and the pH solution reading will further give you the data of the pH, presence of pH in the solution, or we can say in the sample. The calomel electrode terminal is slightly sensitive and would be damaged if it is direct in contact with the blood. To protect that thing from the damage, it is separated from blood samples by creating a salt contact bridge. This salt bridge is typically a potassium chloride, that is the KCl solution, that is separated from the blood by a very thin membrane. The calomel electrode terminal is used because it functions best with KCl. The salt bridge may also be called as the liquid junction. Another electrode we are dealing with is the SANS electrode. Although blood could be sampled in the pH electrode, this particular configuration presents several problems. This system would require a large blood sample volume. Furthermore, the blood would not be at body temperature and would be exposed to the air. The motor compact of the pH electrodes, that is in terms of SANS electrodes, facilitates pH measurement at 37 degrees Celsius with a small blood sample and is accomplished by drawing the blood sample into a pH sensitive glass capillary tube. A membrane at the end of the capillary tube then connects the blood sample to a large reservoir, liquid junction, and the calomel half cell. Now we will gonna see about the blood PCO2 sensor. The electrode function, the PCO2 electrode is modified version of the pH electrode. That is, the structure is same, function is same, but it is a modified version. In the PCO2 electrode, however, blood does not come in the direct contact with the pH sensitive glass. Rather, the blood comes in the contact with a CO2 permeable membrane. The membrane may be made of silicone rubber tube, Teflon, or similar substance that is readily permeable to CO2. On the other side of the membrane is bicarbonate solution that is in direct contact with the pH sensitive glass. The bicarbonate solution is also in contact with a silver chloride electrode terminal. Thus, the PCO2 electrode actually has two AgCl electrode terminal within it. And here no salt bridge is necessary because blood is not in direct contact with the electrode terminal. If you see the diagram of this sensor for the PCO2 measure, blood PCO2 measurement, it is as same as we have seen in the blood pH sensor measurement. Just the difference is the addition of the silicone membrane, which can be seen on the bottom part of the right side of the tube, and remaining thing is the same, like we are having the again AgCl half cell reference solution with known pH on the left side, right side the bicarbonate solution, and mid we have the common pH sensitive glass electrode terminal. At the top we are having the voltmeter, which is having the reading from the both the half cells. Left side we can see the amplifier to amplify the signal. Okay, and as per that change in the concentration and as per that change in the level of the oxygen CO2 level, you can clearly see the change and the presence of the PCO2 in that solution, which is been further measured by the help of this device. A chemical reaction occurs within the bicarbonate solution as CO2 diffuser syndrome, as we can clearly see in that diagram. This reaction, which is known as the hydrolysis reaction, results in the production of hydrogen ions and a pH change of the bicarbonate solution. The pH change is in the direct proportion to the PCO2. Thus, the corresponding voltage change can be converted into PCO2 reading and reflected on the voltmeter. Swearingen-Gauss electrode, that is one of the type of PCO2 electrode which is used in clinical environment most commonly, that is known as the Swearingen-Gauss electrode. In that, only a very thin layer of the bicarbonate solution is exposed to the blood sample so that the measurement is more accurate as compared to the previous measurement device we have seen. Now, microbial biosensor. A microbial biosensor is an analytical device that couples microorganisms with a transducer to enable rapid, accurate and sensitive detection of target analytes in fields as diverse as medicines, environmental monitoring, defense, food processing and safety. The earlier microbial biosensors used the respiratory and metabolic function of the microorganism to detect a substance that is either a substrate or an inhibitor of this process. A microbial biosensor is a systematic gadget that immobilizes microorganisms onto a transducer for the discovery of the target analytes. Microbial biosensors have turned into a standout amongst the most valuable methods for checking natural nourishment and clinical specimens. Now, enzyme-based biosensor. The biological material that is enzyme is immobilized on the transducer. The analytes, that is substrate, binds to the enzyme to form a bound analyte. The analyte is converted into a product which could be associated with the release of heat, gas or hydrogen ions. The transducer that converts the product link change into electrical signal, which is amplified and measured. On the right side we can clearly see the diagram representation of that. Our individual is taking the blood glucose test, is adding the blood in the strip. It is one of the example of enzyme-based biosensor. In that, after adding the thing, the analyte will bind with the enzyme and found a bound analyte which is present in the strip, and that analyte will be give the change in the product level associated with the hydrogen ion change on the strip, and that change is further be transmitted and analyzed and converted by the help of transducer linked with the processor, and it will be given you the measurable signal to the device, and that device will give you the appropriate reading of the blood glucose. Okay, So the procedure of a blood glucometer is an electronic device for measuring the blood glucose level. As we can see in the right side diagram, it's all about the blood glucose, how it's measuring. Okay, in that a relatively small drop of blood is placed on a disposable test strip which interfaces with a digital meter. Within seconds, the level of blood glucose will be shown on a digital display. That is how we are using the blood glucose meter in the digital blood glucose meter nowadays. The working mechanism behind that, when the glucose react with the glucose oxidase GOD to form a glucolic acid. The glucose mediator reacts with the surrounding oxygen to form H2O2, hydrogen peroxide. And the higher the glucose content, higher the O2 consumption. And glucose content can be detected by the electrode. As we can see on the right side of the image, we are having O2 interacting with the glucose oxidase and forming the H2O2 on the electrode surface, and that is being further giving you to the gluconic acid, and that is being measured by the electrode. Different types of transducer can be used for that enzyme-based biosensor measurement. Very first in that, oxygen sensor that measures O2 concentration, a pH sensor that measures the gluconic acid production, a peroxide sensor that measures H2O2 concentration. These were some different types of transducer which is being used for the measurement of this one, enzyme-based biosensors. If we gonna see about the types of biosensors, in that we have seen in the very first in slides, we can clearly see measure the change in the heat. We will gonna use the calorimetric biosensor. If you want to measure the change in the optical scenarios or in the change in light sensitivity, we will be using the optical biosensor. If you want to measure the change in the electrical conductivity, we will be gonna use the electrometric biosensor. If you want to measure the change in mass, you will use the piezoelectric transducer. If you want to measure the change in charge, you will use the potentiometric transducer. The calorimetric biosensor, these are called as a thermometric biosensor or thermal biosensor. Many enzyme catalyzed reaction produces heat, exothermic heat. The calorimetric biosensor measures the change in temperature of the solution containing the analyte. Optical biosensor. This biosensor measures the change in fluorescence or in absorption caused by the product generated by the catalytic reaction. Potentiometric biosensor. These biosensor uses ion selective electrode to convert the biological reaction into electronic signal. The electrode employed are most commonly pH meter glass electrodes. The advantages of using these potentiometric biosensors are having the most specific than cell-based sensor, faster response due to shortage diffusion path, that is there is no cell wall, that's why the response is fast. The disadvantage of having this type of biosensor is it is more expensive to produce due to the additional problem of isolation of the enzyme, and the enzymes are more of often unstable when isolated. Example, we can see this is the blood, all blood glucose measurement. It is coming in the enzyme based biosensor, or all of its test we are doing with the blood type strips. Those all comes in this enzyme based biosensor. So in this chapter we have learned about the chemical and enzyme based biosensor, how the working of the biosensor is going on when it is dealing with the terms of chemical and enzyme. We have seen the blood gas sensors, we have seen the blood pH sensor, we have studied about PCO2 sensor, microbial biosensor, and we have seen the enzyme based biosensor. These are the reference which are used in the preparation of these slides. You can go through all these references for having more detailed study about the chemical and enzyme based biosensors. Thank you for listening. Hope to see you in the next chapter.